Good morning, or good afternoon at this point. It's a pleasure to be here. I'm Josh Denny, and I'm going to talk through a couple cohorts and how I think that they could enable or facilitate clinical trials. And the first, I'm going to focus on the All of Us research program, which is a program I'm sure most of you have heard of at this point. It began as the Precision Medicine Initiative cohort program, and the core group was put together in July of 2016, and we launched nationally May of this year. The goal from the beginning was at least 1 million diverse participants, and we have made a lot of progress in that front. The core way to recruit is through two avenues. We have more traditional route would be healthcare provider organizations, which would be academic healthcare systems or other healthcare systems that would recruit through their patients and enroll them. And then to enable people to enroll from anywhere, we have a route called direct volunteers, which would enable them to sign up digitally and then be connected to a number of other providers that potentially could do an in-person visit. And so we want to collect the same sort of information across all individuals, which includes a common process for enrollment and consenting, including sharing your electronic health record data. That data is centralized, as is all the other data, and common interfaces are used for all the recruitment. A series of health surveys in different modules, each about 10 to 15 minutes long. Baseline measurements, which are fairly limited in all of us. Height, weight, BMI, waist and hip circumference, blood pressure, and pulse. And then urine and blood, and if for some reason we can't capture blood, saliva. And then smartphones and wearables are part of this, we envision as well. I mentioned we launched in May, and we currently have 83,000 full participants that have completed all elements. A little over 145,000 have signed up on the website and are in some process here. We're particularly, I mentioned we're focused on diversity, and we have over 45% that are non-white. Over 75% represent are what we call underrepresented biomedical research, which looks at sexual gender minorities, rurality, a low socioeconomic status. A key part of our program is participants will get data back, including such data as their genetics and their electronic health record data, and that they are recontactable. It's a longitudinal process. This just shows our planned enrollment sites. All elements of the pluses, which represents the healthcare provider organizations, have launched at this point. We have over 200 sites that are enrolling now, and we plan to continue to enroll more. With the dots, you see some of our other partners for direct volunteers like Quest, EMSI, Walgreens, these and the blood banks, and these groups will be are still very much in the process of being scaled up across the country. The overlay there in gray is the population density, so you can see they follow. I'm an informaticist, so I'm going to show one sort of dense schematic. The most important part of this, though, is that data is coming from a lot of different resources and being centralized. We're harmonizing to a common data model. We use OMOP, which has been adopted by a number of others internationally, and really trying to capture the full healthcare data from the HPOs. We are adding data centrally. That is really in its infancy. We're looking at capturing data from direct volunteers through a number of avenues, health data aggregators, health information exchanges, companies like Picnic Health and others we've thought about. And then there's protocols called Sync for Science, which has been around from the beginning, that we're using to enable individuals to share their own records, and that's in pilot too. I'll talk a little bit more about that later. All this is harmonized against the health surveys that they're filling out and the biospecimens in our own raw data repository as we move to curate the data and make it available for research, which includes removing things like the obvious patient identifiers from the healthcare data. I'll tell you that version 1 is very much well underway. We have around 49,000 EHRs that have been shared, and some of those are starting to also send us clinical narrative data as well. So we're making good progress in that, having just started a few months ago. I mentioned Sync for Science, and this is a strategy that's based on FHIR. I heard that mentioned before, and it's a structured XML form of sharing data, and it allows you to go from a research app to a patient portal and authenticate for that patient portal to share data back to the research app over time. Similarly, the same sorts of technology can be used on your smartphone, and this is just a snapshot from my phone and my health record, so you can all see my cholesterol values. That many places have implemented now, whereby you can use basically the same structure of using a fire report to sync with other devices, and Apple's partnered with a number of others, and you can imagine that continuing to expand. We hope it continues to expand. I want to emphasize that the things like clinical trials have been on the roadmap for all of us since the beginning. I just marked a few here that have of the scientific priorities that have been that relate here. Certainly, we think of all of us enabling 3D contact, a future engagement with participants that they could, if they so choose, be made available for clinical trials. And you could imagine that there could be a much smarter targeting of individuals and what trials are actually relevant to them in their choosing of whether they would want to participate. In addition, I think there's a great opportunity from resources such as all of us and other resources to be smarter about maybe what we pursue and think about health disparities and other elements in those things. So I was asked to talk a little bit about a few other resources as well. So I want to mention UK Biobank. These slides are modified from Kathy Sudlow, who's the chief scientist for the UK Biobank. The UK Biobank recruited 500,000 individuals between 2006 and 2010, initially ages 40 to 69, and they consent for really all types of health researcher from a broad range of researchers, and that they can have data linked over time. And they use extensive baseline questionnaires where they come into the trailers across 22 recruitment sites and measure a number of things, and do things like electrocardiograms and basic biological assays, as well as collecting biosamples. And it's open for access through an application process. And then currently the model is you would download the data once approved and can do analysis. Looking at the assays done in the UK Biobank and in all of us, what's being planned, UK Biobank currently has GWAS data on all their participants. They have a standard panel of biochemical assays like lipids and metabolites, and they're underway with working on whole exome sequencing. It's progressing. They're starting whole genome sequencing, and a number of other assays that cover metabolomics, and then plans for proteomics. They also have whole body imaging and activity monitoring that are in progress across subsets of the cohort. And all of us in 2019, we're looking to start GWAS on the whole 1 million, as well as starting whole genome sequencing. Obviously, due to the technologies, it's faster to get GWAS going, and we expect one of the reasons we're doing both is we can deliver that faster and also return data to participants faster while we scale up the whole genome sequencing effort. There's a small assay pilot that we're looking at as well, and we may do other clinical labs. We have a planned Fitbit pilot. We're building technologies for people to link in their own devices, such as Fitbit will probably be first, and then Apple Watch and other things. And we're looking at GIS data linkages with EPA data, NOAA data, things like that, and many other tests are in consideration. There are a number of other projects here. Another one that has received a lot of press and is enrolling is Project Baseline, which is a joint venture between Verily, Duke, Stanford, and Google. Their goal is to recruit around 10,000 people. It's very intense assessment and highly digital. All of us is more digital than the UK Biobank. Project Baseline is more digital than all of us, with a number of sensors that they're putting in homes and the depth of the in-person assessment. They also capture bias specimens and surveys as well. So a lot of our work has been in electronic health records. I use this slide to emphasize at least at one academic medical center, the amount of data you can get from the EHR, and how even if someone enrolls, in this case starting in 2007, you have a lot of data retrospectively, and sometimes decades of data retrospectively that you can use essentially in silico for relatively inexpensively. There is work to be done, but the data has been accrued relatively for free for the research project. So if you start that and think about adding your smartphone data and your surveys and other kinds of things that we're going to get from these cohorts, what can we do with this? I think one of the use cases would be phenotyping and doing genomic investigations to identify new drug targets and think about understanding pharmacogenetic discovery. and then doing phenome-wide investigations would let you think about, what kinds of associations you can get with a given genetic variant. So if you know a target for a drug, then you can look at the genomic variation in that drug and think about what potential indications you may have, as well as potential adverse effects. And so this could lead towards, directing what kinds of outcomes you want to look at, and also prioritizing maybe what kinds of indications would be pursued, and when you think about things like repurposing as well. And then redefining disease is another thing we think about, using artificial intelligence and other types of clustering approaches, and how we can think about subtypes of disease. And this gives some examples of people with elevated uric acid and autism. Autism's on the left, and the clusters there that can be identified by reclustering patients. And then those may link to certain outcomes in genetics and things like that, which could target towards subtypes, subpopulations that you want to treat. Here's one example of an early study we did looking at rheumatoid arthritis that started with a simple GWAS on a time which was a huge population of around 100,000 people. There are many much larger genetic studies now, but we accrued about 27,000 patients that had RA and then over 70,000 controls, and we identified 101 genetic loci for rheumatoid arthritis. Many of these have tiny effect sizes, as with every GWAS. But what's exciting about this is when we looked at the regions that were identified by the genetic loci, about half of these were new. Most of them, or at least many of them, point to active treatments for rheumatoid arthritis. And so this is large-scale validation, sort of suggesting that you can use genetics to recapitulate drug targets, and it also identifies potential other targets that could be treatments as well for rheumatoid arthritis. And then other studies have gone through and shown similar such effects on larger scale. Here's an example with looking at drug adverse effects. First, looking at clopidogrel, which is a prodrug. It's activated by CYP2C19 to its active form, and so individuals who have loss of function variants for CYP2C19 are more likely to have adverse events. Clinical trials and retrospective mining of those showed about a 50% increase of stroke, MI, or death with loss of function carriers. And when we looked in an electronic health record, we basically saw the same effect size, about 50% increased risk of adverse events. And that's one example of discovering pharmacogenetics and maybe thinking of those subpopulations once a drug is out in the wild. And then I imagine most of this room is probably familiar with the story of PCSK9. When you look at diverse populations, identifying, in this case, people of African ancestry who had very low cholesterol and have loss of function variants in PCSK9, were found to have also not only lower LDLs, but also have dramatically decreased risk of heart disease. And then this led to, a number of drugs on the market which do dramatically reduce cholesterol levels on top of statins and do reduce heart disease. I like this example because it required a diverse population to find the effect. It also shows genetics leading to a drug discovery. But what would be the potential adverse effects? So I mentioned the idea of using a phenome-wide study to study for adverse effects. This is just looking at one outcome. So using genetics now to look at the outcome of potential diabetes risk in people that would be treated with PCSK9 inhibitors, using genetics to recapitulate that effect. And you can see that using genetic variants would predict an increased risk for diabetes with PCSK9 inhibitors. And we've looked at other associations as well, some of which have been looked at in animal models and have some evidence there as well. So in summary, I've given a few examples of how I think these observational cohorts and a growing landscape of them can help accelerate our clinical trials. One is through basic discovery, which is really where most of these cohorts probably start. But some of these also enable direct recruitment and can facilitate assessment via their infrastructure. So if you think about all of us and the recruitment centers and that picture I showed, this is infrastructure that could enable for assessment, and having that within a population treated for something could enable more cost-effective recruitment and follow-up. And some of the technologies like Sync for Science are available to anyone, not just an existing cohort like all of us. And then faster recruitment for targeted clinical trials. For instance, if you want to find a population of cystic fibrosis with a specific genetic variant, those those populations would be embedded in these resources that are already genomically sequenced, for instance. And so that sub-targeting of populations, if they're found, may be sped and may be more, may enhance recruitment in that way. And then I think that there's some evidence to enhance trial design. For instance, with PCSK9, the genetics, one of the stories I didn't show was potential for neural tube defects. So if that's the case, then maybe that sort of study could inform the research that is doing in the clinical trials in that, especially if you're looking at, if in that case, looking at pregnant populations. And really emphasizing what's the most strongest hypotheses. So the cohort landscape's expanding. I gave just a couple examples. There's also groups like 23andMe, PCORNet, we'll hear more about. There's a number of other resources out there, like the Million Veterans Program. Some of these, are more available for discovery. Some of them also have recontactability. Some of them will probably evolve to have recontactability. And I think that there's emerging opportunities that will happen over time that could help this to grow as a richer resource, hopefully to enable some of this. So I'll just end with a couple of pictures of folks that really contributed to all of this. The first All of Us network meeting in 2016, and I lead the Data and Research Center with a couple of colleagues at Vanderbilt for All of Us. So their picture as well, but of New York skyline, not Vanderbilt. We don't have that many tall buildings yet. Thank you very much.